Hi, everyone. My name's Alyssa,
And I'm Rachel.
and we're on the executive committee of the Western Biochemistry Club.
So we're here to tell you a little bit more about our club today. Well, I guess actually I'll introduce myself first. I'm a third year in Med-Sci with honors spec in biochem and cell bio. And
I'm in my third year of medical sciences as well, doing an honors specialization in biochemistry and cancer biology.
so last year we were also in your position taking this course, except it was online. And it could be feeling very overwhelmed, especially after first year. This is a lot of content, but do not fear because there is the biochem mentorship program that's here to help you. So basically how this works is that we have weekly review sessions. And also we have a Facebook group page where I think they will be making one. And you can post your questions, any concerns about the material that you have, and the mentors will always be available to jump in and answer any questions or concerns. We also have review sessions closer to the midterms or finals, and look forward to those.
You can join our club for $10 membership. If you do that within the next week, I believe you'll be entered to win one of these fancy t-shirts we've got on. You also have access to events such as trivia nights, bar nights. We also host a really cool event called Proteins with Profs, where you have the opportunity to sit down with a prof, usually over a meal, and ask any questions you have, whether it's about research, whether it's about courses, whether it's just about your future next steps. And you can do that. We're on Instagram and Facebook under Western Biochemistry Club.
All right, thank you so much.
All right, thank you. So yeah, it's a good program. It's upper year students here to help you because, as they said, they know what it's like to be in this course. And yeah, so and I have one of those t-shirts. They're pretty good. Okay, so howdy. I'm not Dr. Bofa. Sorry to disappoint you. I am Dr. McLaughlin, and I am taking over the course for the next little while. I have some similarities with Dr. Bofa. We both have a nice salt and pepper beard, if you can see it through the mask. We both have daughters who attended Western. And we share other things in common that you might be more interested in. And that is our approach to testing. Neither one of us likes multiple choice questions. So we tend to avoid those on exams. So that's one thing I'm sure you'll want to know. The other thing is our approach to the testable material. So as Dr. Bofa said in his section, the same thing applies to mine. What you're responsible for the test is what I cover in the lecture and what's in the course notes, right? Okay, so if you have a good handle on that, then you're good to go. So if you read something in the textbook and some detail of something I didn't talk about in class or I didn't emphasize in class, it's okay. Don't worry about it. I mean, it might be worthwhile to read to expand your knowledge and your understanding of things, but I'm not going to specifically test you on these details that you find in the textbook that are not in the notes and I didn't cover in lecture and are not on the lecture slides, okay? And so in this regard, when you're studying, I would encourage you to look at the learning outcomes. We have written learning outcomes for each topic about things you should be able to do. And you should be able to link every question on the exam to one of those learning outcomes, okay? So if you are studying and you're reading those learning outcomes and you read one and you say, oh, I don't think I can do that, well, that's something you should look into. But if, you know, the night before the test and you're like, oh, I can do all these learning outcomes, I feel good, then you should feel good because so students always ask me, is this on the test? Is that on the test? Go to the learning outcomes. And if you can do the learning outcomes, then you should be prepared, all right? So, now, my office hours is one difference between us. My office hours are on Tuesdays between 2:30 and 4:30, as it says, in my office, which is Medical Science Building 349. I will offer to meet people on Zoom. If you want to connect with me on Zoom, just let me know. And if nobody lets me know they're going to come to my Zoom office hours, I just won't create a Zoom thing for that, okay? But I encourage you to come in person if you like, or through Zoom. Just let me know ahead of time if you want to do the Zoom thing, okay? Now, I get a lot of emails. And sometimes students email me things about the course material. And I would say that, it's not a bad idea to reach out to the prof to get help on the course material, but the email is not the appropriate format for that. Because as I say, I got a lot of emails. It takes me a lot of time to go through emails, and I'm, a very busy guy. And because I have a lot of deep things to ponder as a professor. We think deep thoughts, and we're always wrestling with deep questions. Like, what did that guy do to Olivia Rodrigo anyway? And will she ever be able to get over it? I don't know either. And will she be able to parallel park ever? I don't know. These are things I wonder and grapple with while I'm sitting in my office. So that means that consumes all my time. So I can't respond to individual emails about course content. So if you have questions about the course content, please post them on the forum, all right? And we've been pretty attentive, I think, on the forum and answering questions. So please do that, and that's the best place. If you have something, you want to discuss that's related to you personally, that's not for public consumption, of course, you can send me an email about that, and I will read it. But for course content stuff that everyone would benefit from, I will respond to those on the forum, and that's the best way to go with that, to deal with that. Now, Tasnim, as we said, is available for an appointment. You can always contact Tasnim to get her opinion on things or to get her help with course material. Okay, and now the other difference between myself and Dr. Bofe is, one of them is that I tend to ask more clicker questions. Now he has used it, but I tend to ask more. So here's one to get you going. And if you got the app, you just open the app, or you can go to iClicker.com and you should be able to find this course 2280. And if you're in 2288, just click 2280. So just in general, why do you think it's important to study biochemistry? I gave some possible reasons here, but you might have another reason, so I gave you an option for that. Okay, we got some pretty good responses. I want to see what you think about this. So I'll give you a few more seconds to put an answer in. Let's see what we got here. Okay, so most people, sort of the basic research answer, understand how things work, but also develop medical treatments. Yeah, and a few people picked other things. That's cool. Yeah, so there's different reasons to study biochemistry, and you might think more than one of these is important, right? So I asked you for the most important one, but you could have different reasons. And these are some reasons that I thought, I kind of figured actually human disease would be first, but you guys like the quest for knowledge. That's fine too. There's other reasons to do it, but there's one reason that you might consider the most important reason to study biochemistry. It's not on the list, and that is to win the Nobel Prize. Which is obviously a very well-known prize, and you get very famous for doing that. And again, as I sit in my office, I think about these things. If you do an analysis of the Nobel Prize categories, there's 6 categories, right? And then you can ask yourself, what are the odds of a biochemist winning in these categories? And if you go down the list, you'll find that in fact, many biochemists have won in either of these last 2 categories, right? So why would you study physics, really? Because you only have one shot, right? If you study biochemistry, you double your chances, right? So, but I should say that there is one biochemist who won the Nobel Peace Prize, and that was Linus Pauling. So I have to give a shout out to him. He won 2 Nobel Prizes, one in chemistry and one in peace. And that deserves a picture. So here he is right there. He's with the alpha helix, which is something that he sort of dreamed up and theorized about with this partner here. So his partner here, Dr. Corey, understood in the 50s what a scientist was supposed to look like. Sort of nerdy looking guy with a white shirt and a tie, and Pauling doesn't fit the mold, right? So he broke the mold in a lot of ways. Anyway, just shout out to Linus Pauling for the Nobel Prize. Now, I will tell you one thing. I am never going to win the Nobel Prize. I know that at this point in my life. I'm old enough to realize that this is not going to happen. However, I've been teaching this class for a long time. And there's a lot of smart students in this class. Look around. There's smart people in this room, right? And I think the odds are pretty decent that somebody going through this class is going to win the Nobel Prize someday, okay? That's my faith in you. This is my confidence. And as the longer I teach this course, the more and more students I've been teaching, the better chance that someone in this class is going to win the Nobel Prize. So if you win, or when you win the Nobel Prize, please do me one favor. Mention my name in your acceptance speech. Okay, I've been asking the class to do this for years now, and I think it's going to happen sooner or later. Someone is going to mention my name in Sweden, and then I'll feel gratified. Okay, so that's a little thing. So now I will ask the clicker questions, and some of them will be for fun, but a lot of them will be more serious. And the point of that is to help you think through the things in class, and I'll ask you to talk to your neighbor. You might get a discussion going about it. It helps you assess how things are going for you, right? And if you get them wrong, it's okay. It's normal to not be perfect, right? We're human beings. We're not going to be perfect. Making mistakes is very often a great way to learn, because then you're poised. Okay, what did I get wrong here? I can learn something new right now. Okay, so if you get a quick question wrong, don't get upset about it. Just pay attention to what I'm about to say next, because it probably means you're going to learn something. But I'll say that I was a student, and sometimes I got discouraged. And if you get a bunch of these wrong, you might start getting discouraged. And if you're studying, you might be reading and thinking, "Wow, this is hard, and I can't seem to get it." And you might start to think, well, I don't even belong here. Well, I don't even belong in this class. I don't even belong at Western. What am I doing here? So here's my word to you, if you can relate to this. You belong here, okay? You got into Western. It's not easy to get into Western, and you got into this class. Western thinks you belong here. I think you belong here, okay? There's all sorts of people here. Everyone in this room is capable of learning biochemistry and succeeding in this course. All right, so believe in yourself, but use the help. So, and I'm here to help you. Dr. Bofa, he's here to help you, and the TA are here to help you. The mentorship program here to help you. Use the supports. Okay, so if you find yourself getting discouraged, don't give up. It's all right. Just figure out how we can help you to get where you want to go. Okay, so hang in there. Because yeah, it's not always going to be easy, but that's the university. If this university education was easy, it wouldn't be worth that much, right? You got to work for it, and when you do the work, you get what you came for, which is an education. Okay, so there I'll stop philosophizing for a bit, and we can start talking about things. So I don't have molecule today, but I do have comics. Okay, so I don't know what you prefer, and don't answer that, but this is what I do, because I'm a fan of Calvin and Hobbes, which new Calvin and Hobbes were being made when I was a teenager, if you believe it. And I always just go down to the newspaper. We had those things in those days, and look at the Calvin and Hobbes. So I like to pull out comics that relate to the things that I'm teaching, or at least in my mind they do. So here's a image of Calvin. Now it's a nice sunny day today, and we don't have any snow, but snow is coming. Winter is coming, as they say. And so he's building the snow fort, but it turns out he's built it across the driveway, and his dad's going to have a hard time getting out. And so this reminded me of maybe how some people might think of a membrane which separates the outside of the cell from the inside as some sort of impermeable barrier. But of course, membranes are more than just that. And in fact, it's not true that they're impermeable. You have to get things across. And throughout the course, we're going to talk about moving things across membranes at various points. But in topic 6, we're going to talk about membranes and lipids and focus on them. So here's this course map thing. You've already done Unit A, where Dr. Bofa's talked about proteins. Now we're getting into Unit B, which is broadly labeled metabolism, but I'm going to talk about lipids and carbohydrates, and then we're going to get into energy metabolism. And you can loosely subdivide this into different sections. If you look at the course notes, you could probably find these 4 major themes running through them. Okay. I'm not going to reproduce learning outcomes, but I'm just going to, in bullet point form, there's 8 learning outcomes for topic 6. They deal with the different classes of lipids, what those lipids are doing in living systems, what bilayers are like, their fluidity and their properties. We're going to talk about proteins, how they associate with bilayers. Very little bit about studying membrane proteins, what membrane proteins are doing in the membrane, and then how you get things across the membrane. Okay, so that's where we're going. Again, pay attention to those learning outcomes. They'll help you organize your thoughts around topics. Okay, but before we go further, I'm going to remind you of these major classes of biomolecule that Dr. Bopha talked about. He spent a lot of time talking about amino acids, which are joined together to make proteins. And now we're going to launch into lipids. The next topic, we'll talk about carbohydrates, and then later, after the midterm, we'll talk about nucleotides, okay, and nucleic acids. All right. Now, before we go further, I'm going to introduce you to Saurabh, who was a former student in this class. He used to sit right over here. And he let me use his picture and a number of things. He took some pictures, and here's Saurabh. Saurabh, it's been a while, so if Saurabh's not in the latest fashion, give him some forgiveness, because these pictures are not, I just say that in his benefit. He is a real person, okay? And I'm going to tell some stories about Saurabh, but I don't want you to think that stories I tell about Saurabh are true. He's a good-natured guy, and he's actually a medical doctor now, and he's let me use his image and tell stories about him just for that reason. So Saurabh likes to cook, and he's a pretty good cook, and he showed me an example of this as some meal that he liked to make. But every now and then, you get busy, and you don't have time to buy groceries, or you're just on the run, and you feel like, you know what, I don't have time to cook today. I'm going to grab some fast food. And it's probably not the most healthy thing to do, but every one of us does it now and then. I think most of us do. So Saurabh one day is too busy. He's coming to class, going to class, doing stuff. He decided to stop off at McDonald's for a Big Mac meal. Okay, so Saurabh, he's hungry, right? He's got to feed that brain. So he sits down, and he's all excited to eat his Big Mac meal. There he is, and he's got his bag there, and he dives in, and then he polishes it off in a hurry, because he's in a hurry, and he starts to wonder, what have I done? What did I do to myself? So then, you can go to McDonald's website, and you can look up nutrition information. So let me ask you this. He's eating this meal to get energy. I talked about these 4 classes of biomolecule. How many of these are going to be providing energy to Saurabh as he goes about his busy day? And I should say the clicker questions are optional, right? You don't have to do them. You don't get marked for them. No, if you don't do them, there's no problem. But I think it's fun to do them, yeah. No, Saurabh has friends. Okay, it's been a minute and we got lots of responses. Let's see what people think. Okay, so most people picked D, which is 3, but some people felt 4. All 4 of them would provide, and some people felt only 2. So the answer here is that is actually 3. 3 is correct. That all of these provide energy except for nucleic acid. Nucleic acids, generally speaking, our bodies do not break down to obtain energy. And this is sort of a approximation of how much energy you get per gram of each of these different types of biomolecules. So, and this is covered actually in topic 13 in the notes, so don't worry, it's on the notes for topic 6, but I'm trying to bring it, I don't know, I'm doing a foreshadowing thing here. So you get about 9 calories per gram of fat that you eat and about 4 calories from the carbohydrates and the proteins. And as we go through the metabolism section, I hope you'll be able to connect what we're talking about in metabolism, energy metabolism, to the actual food that you eat and the stuff that you're putting into your bodies, okay? But I have a question now, before we go further. Saying calories per gram, does anybody know what do we mean by a calorie? You're all science students here, you've taken some science. What's a calorie? How do you define a calorie? Anybody? Yeah. Okay, it says the energy needed to increase the temperature of 1 gram of water by 1 degree Celsius. And she's nodding. Does that sound like a reasonable definition? Yeah, people are nodding. Okay, very good. Good. You guys did take previous science. This is good. So we want to pay attention to the capital C calorie and the small c calorie, because he gave me the definition of a scientific calorie, the small c calorie. But if you read nutrition labels and stuff, you'll see capital C calorie, and that's basically the scientific calorie times 1,000, right? So that's the amount of energy needed to raise the temperature of 1 kilogram of water by 1 degree. So when I say 9 calories per gram, that's 9,000 scientific, 9,000 scientific calories. Okay. So just get that in your head. But basically a calorie is a unit of energy, right? It's something we can use to power ourselves. And we'll talk about how that works shortly. So, or through my topic. So now here's a question. There's 3 major components in a Big Mac meal. You got the Big Macs, you got large fries, and you got large Coca-Cola. In this case, that's what he chose. So which one gives them the most energy? I don't really expect you to know this, but you could take a guess. Okay, a few more seconds. Okay, so most people felt it was the Big Mac, and the Coke came in second and the fries third. So you can go online, of course, and look this up, right? And so, you know, I encourage you to do that because it sort of gives you a perspective. But so I did that just for class, and this is what I found. So indeed, it's true. The Big Mac has the most energy, but the fries maybe has more than you thought it did, okay, on the large fries. And the Coke comes in a distant third. And so you'll see when you look at nutrition labels, you will see these different subcategories, right? And you'll see that they list those 3 major classes of biomolecule that I just talked about that are providing energy. And it tells you how much there is of each. And then they further subdivide the fat and the carbohydrates typically into these other categories. And so you'll see then that both the Big Mac and the fries have a fair amount of fat and a fair amount of carbohydrates. But then the Big Mac wins on the proteins, and that really pushes it over the edge. Whereas Coke has just the sugar in it. But if you're very astute and good at math, you may have noticed that, you know, it says 32 grams of fat, and then it tells you 10 grams of saturated fat, 0.5 of trans fat, and 70 milligrams of cholesterol. whoa, that doesn't add up to 32. What are they hiding from you? Like what? There's not everything is given there. And besides which, it doesn't really give you a definition. What is saturated fat? What is trans fat? So hopefully by the end of my section of this course, you'll be able to interpret all this stuff and understand what's missing from these labels. And so that's, and so, yeah, again, just to preempt the question, no, I'm not going to ask you on the exam how many calories are in a Big Mac meal, okay? This is just making a point, okay? So stay with me. So now we're going to talk about fat structure. We're finally getting into the lecture, 25 minutes in, about fatty acids. So fatty acids are a major class of lipids, and I should actually start out by defining what do I mean by lipid. A lipid, it's a bit hard to strictly define, but you could define it this way. It's a biological molecule that is not very soluble at all in water. Okay, that's true of all lipids. They're biological molecules made by some organism, and they're not very soluble at all, if at all, in water. Whereas most biological molecules are water soluble. And so now I'm just going to talk about several, I'm going to divide them into different classes based on their chemical structure. And you are expected to understand the different structures, the different classes that I'm talking about. So first, we're going to start with fatty acids. These are used as a source of energy, as we've just seen. They're also used for structural purposes, as we will see. Okay. Now, here's an example of a fatty acid. It's an acid, right? It's got a carboxylic acid group, but then it's got typically a long hydrocarbon chain, which is composed just of carbon and hydrogen, right? So you've seen these types of molecules in organic chemistry. And this particular one has, I think, 16, and they tend to have even numbers of carbon atoms in them. You can have fatty acids with an odd number of carbons, but as a rule, most of them are made and have even numbers of carbon atoms. And they can range in different lengths. You know, really the shortest one might be, you know, acetic acid, which only has 2 carbons. And you can get them very long, like 24, 28, even 30, okay? They can be quite long in the number of carbons. Now, saturated, what we mean by that is that you can think of it this way: they have as many hydrogen atoms attached to carbon as you possibly could have, okay? So all these carbons, you know, the idea is, again, this is sort of a convention of organic chemistry. Each point here is a carbon atom, and they're connected by these lines, which are the single bonds between carbon atoms. And you have to assume then that there's hydrogens attached to the carbons, and they're not shown in the structure because that would just clutter everything up. But basically, a saturated fatty acid is fully saturated with hydrogens. It's got as many hydrogens as possible on the carbon atom. And you can contrast that then with an unsaturated fatty acid. And these are monounsaturated fatty acids because they have one element of unsaturation. That's essentially this double bond. And so there you've removed some hydrogen and made more than one bond between these 2 carbon atoms. Okay, so that means that you could put more hydrogens on there if you were able to, or you wanted to, but it's unsaturated because it's not, doesn't have all the hydrogens. Now, there's 2 different types of double bonds that you can make, trans and cis, and hopefully you can see the difference between this. Again, if you've taken organic chemistry, you probably get the idea. The trans double bonds, the carbons, are on opposite sides because this bond here with the hydrogens and the carbons, it forms a plane. Okay, and so in the trans, you've got them on the opposite side, and on the cis, the 2 carbons are on the same side. Okay, and then you have a hydrogen up here and a hydrogen up here. So that's just the nomenclature we use. Trans is on the opposite side, cis is on the same side. Now, it turns out that in living systems, we mostly make cis double bonds. In fact, it's pretty rare at all to see a fatty acid in nature with a trans double bond. So cis is the bond that's favored in fatty acids, and the double bond is added at a specific position. It wasn't random that I put this double bond right where it is. This is between the 9th and the 10th carbons from the carboxylic acid end. And that is typically where the first double bond will be added to a fatty acid. Fatty acid gets made as a saturated fatty acid, and other enzymes come along and they can add double bonds. And the first one will practically always be between this carbons 9 and 10 away from the carboxylic acid N. Okay. Now, this is not the most accurate way to depict the structure because, as I said, this double bond is planar. And you know, double bonds, there's no rotation around the double bonds. So what happens is this actually makes a bend or a kink in the fatty acid chain. And this will become more important when we start talking about membranes. Okay. But it's often, you know, convenient to represent it like this because it takes up not much space in say, a line of text. But just keep in mind that whenever you see a cis double bond, that's going to make a bend in the chain like that. Okay. But as I said, the cis double bonds are pretty much the common ones. So we're going to get rid of the trans guy there. And now we're going to go on to think about, well, you could add more than one double bond, right? So then you could have a polyunsaturated fatty acid with more than one double bond. And I'm showing that here. And again, I'm trying to show you the change in direction that happens with the chain when you add that. Now you'll notice also, again, I didn't randomly put the second double bond in. It's in a very specific spot. It is, there's a carbon in the middle here that has just single bonds to the other carbons around. And then the double bond appears after that. So if you had a double bond, and just for the sake of the people on Zoom, if you had a double bond in this location here, the organic chemists would call that this is a conjugated double bond, right? The pi electron system is you know, ends up being sort of almost connected, especially if you have a benzene ring, right? That's conjugated. That's not the case for these fatty acids. There's always pretty much always a carbon like a CH2 with you know, sp3 orbitals, if you're into that sort of thing. So these double bonds are not connected in that way. Okay, and so that when you add double bonds in a living system to a fatty acid, you're going to tend to separate them in this way. So the third one, and you tend to add them away from the carboxylic acid M first. So if you add another one here, it would be between these two carbons there. Okay, but that's polyunsaturated, meaning more than one element of unsaturation, more than one double bond, basically. Okay. And organic chemists will number these carbons starting from the carboxylic acid N, but in other literature, in particular nutrition literature, they would number the fatty acids from the other end, from the CH3 end over here. And so this gives rise then to this omega-3 and omega-6 nomenclature. So you would call this an omega-6 fatty acid. And the reason for that is because you can count 1, 2, 3, 4, 5, 6 carbons from the CH3 end, and that's where the double bond starts. Okay, so if you hear that, omega-3 fatty acid then would be, you know, the double bond would be 3 carbons in from the end, right? I just want you to be aware so that when you see that, you'll know what it means. Okay, so those are fatty acids. There's a wide variety of structures, right? They tend to be linear, like unbranched in eukaryotes. And they can vary in size, and they can have elements of unsaturation. All right. Those are fatty acids. Now, if you take 3 fatty acids and stick them onto this molecule here, you can end up with a triacylglycerol. So glycerol has 3 carbons in it. Each carbon has a hydroxyl group attached to it. Each one of those hydroxyl groups can make can undergo a reaction with a fatty acid, in which it's basically a condensation reaction where you get an attack on the double bonded carbon there, and you essentially remove water, and you end up with an ester linkage like that. Okay. So you take 3 fatty acids, you stick them on the glycerol, and you get a triacylglycerol. And this is for fatty acid storage. If you have lots of fatty acids that your body doesn't want to use right now, it will put them into triacylglycerols and store them in fat cells. in adipocytes. Okay. And we'll see again as we get through, when we talk about energy metabolism, we'll talk more about triacylglycerols. And I'll point out that I've shown different fatty acids here of different lengths and with different unsaturation. That's okay. You don't have to be the same type of fatty acid in a triacylglycerol. You have variability here. But the name itself is sort of suggestive of the structure, right? Because if you have inorganic chemistry, anytime you've got a group like this here that I've circled in red is called an acyl group. Okay, that's just a definition of terminology. So an acyl group can have various numbers of carbons, but it's basically got a carbonyl, a C with a double bonded oxygen right there attached to something else. That's an acyl group. So in a triacylglycerol, you have 3 acyl groups attached to glycerol, right? So the name implies a structure. Sometimes you'll see these called triglycerides. It's a synonym. It's basically a bit of an older term for the same thing. Okay, so you see triglyceride, that's what they're meaning, a triacylglycerol, and that's used for energy storage. Moving on, if you take a triacylglycerol and instead of having a fatty acyl group on one of the terminal carbons, you add a phosphate, then you have something called a glycerophospholipid. And this then starts to become a type of molecule you'll see in a biological membrane. And we'll talk about this probably in next class, but you've got hydrophobic groups here, right? And again, hydrophobic. This is why, I should back up again. Lipids are not soluble in water, right? And it's because of all these hydrophobic groups, right? You've got CH2, CH3. This is not water soluble. But the phosphate is. This phosphate is hydrophilic. These groups are hydrophobic. So this is called an amphipathic molecule. It's got 2 different natures, partly hydrophilic, partly hydrophobic. That's makes it suitable to form membranes, as we'll see. Okay. And there's a wide variety of glycerophospholipids then. They can have different numbers of carbons, different numbers of elements of unsaturation, or different polar groups attached to the phosphate. You can have the phosphate by itself, or you can have some on the molecule. I just put something called choline on there. You can look in the textbook to get the structure of that. It's a polar molecule. So there's different head groups, they're called, you can have, and this is all the class of glycerophospholipids, right? Okay, so I would expect you on a test to be able to, if I show you a picture of one of these molecules, you'll be able to assign which class it is. Is that a fatty acid? Is it a glycerophospholipid? What is it? Okay. So the next class is similar to the glycerophospholipid. So this up here is one structure. Down here, we've got a different structure. And this is going to be a target question. So I want you to click on the bottom structure somewhere where it differs from the top structure. I'll give you just a bit more time. That's 300, that's good. So, all right. So, not sure what that is. Anyhow, the bottom here got pretty covered, so you can't even see what's going on. So if you look at this structure, there's a nitrogen there, right? A lot of people saw that, the nitrogen there versus an oxygen there. And you can see, oh, there's an ester linkage in the top structure. but not in this bottom structure here. You got this double bond, and there's an OH out here that's not there. So this is the main area that I was looking for people to notice. Okay, so the top structure is a glycerol phospholipid, and the bottom structure is a lot of similarities to that, but a few key differences. And then this is a sphingolipid along the bottom, and it's based on this molecule here, sphingosine. Okay, so sphingolipids are used in membranes just like glycerol phospholipids, but they're not based on glycerol. They're based on sphingosine. So there's one molecule called sphingosine. This is it. Okay, so just like glycerol has a particular structure, sphingosine has a particular structure. It has some similarities, and I sort of drew it in a way that would highlight those. There's 3 carbons here, 2 with an OH group on it, just like glycerol. One has an amine group on it. That's different from glycerol. And then it's got this hydrocarbon chain here, which is hydrophobic, with a double bond. And now you're looking at that, and yes, indeed, that is a trans double bond. And you might be thinking, wait a minute, didn't you just say that in fatty acids you get cis double bonds primarily? And I did say that. And this is not going against that, because this is not a fatty acid, right? This is a sphingosine, and it has no carboxylic acid group. So I stand vindicated. There is a trans double bond in sphingosine. It's not a fatty acid, so it's synthesized in a different way in that sense, okay? So don't worry about that. But so sphingosine is here, and it has these functional groups to which you can add other things. And so you can add a fatty acid chain to this amine group, and instead of getting an ester linkage, you get an amide linkage, very similar to what you see in the peptide backbone, polypeptide backbone. And you can add a long chain of various lengths or various elements of unsaturation to this nitrogen. And now we have a ceramide. And there's a bunch of different ceramides, and they all differ based on what fatty acid is attached to sphingosine. Okay. So when you see reference to a ceramide, they're talking about a molecule that looks just like this one, and it could have a different number of carbons though on this fatty acid acyl chain that's added to the nitrogen. So this is an acyl chain right there. And then you can add polar groups to this final OH, like phosphatidylcholine or phosphocholene, phosphocholene that I just showed on the other. This is a sphingolipid. And this is a sphingomyelin now. And you can see it also has a polar region here and a hydrophobic region here. So it also is a molecule that can form the basis of membranes. And I just want to point out too, you can add sugar molecules up here instead of choline. You can add sugars, in which case you would call it a glycosphingolipid, glyco being a prefix meaning sugar. And there's many different types of sugars or carbohydrates. That's topic 7. We'll get there in a few days. Okay, so, but catch the difference, right, between the structure of sphingolipids and the structure of glycerol phospholipids. They're based on different molecules, so they have slightly different structures, right? And you should be able to tell them apart. Okay, next we get to the 5th class, steroids. And this is quite a bit different than what we've been looking at before. It's, again, fairly hydrophobic molecules, but they're based on this kind of funky ring system where we got, 3 6-membered rings and a 5-membered ring. And the technical name for this is a cyclopentanoperhydrophenanthrene ring system. And I don't say that, again, you don't worry about memorizing that. I just think it's a cool word. So I put it up there. It's useful to know these sorts of words. It can impress people at parties, maybe, depending on the kind of parties you go to. And if you're a poet, it's good to have a good vocabulary too, right? Because if you're writing a poem and you need an 11-syllable word that rhymes with gangrene or something, well, here's your answer, right? So just keep that in mind, in your poetic aspiration. So steroids, a lot of them are hormones. And I've given 2 examples over here, 2 that might be familiar with you from your other knowledge, right? These are testosterone, estradiol. And you can see actually that they're fairly similar in structure, more similar than you might imagine, given their roles. And it's interesting to think that just these small similarities or small differences can cause so many wars between testosterone and estradiol, since, you know, most wars are driven by this testosterone. But so most of them are hormones, and we're not going to talk extensively about hormones. I think I'm back. All right. So as I said, we're not going to talk about a lot about hormones in this course, although they are important naturally. But we will talk about cholesterol. This is a component that you find in eukaryotic membranes, and it is quite hydrophobic, but it does have a tiny hydrophilic group, that OH group there. But essentially, it's a predominantly extremely hydrophobic molecule, and we'll see its role in membranes in a little bit. But here I want to just pause and highlight a scientist who was involved in the efforts that linked originally high cholesterol levels with high blood pressure. And this was found in experimental animals. And so this is Marie Maynard Daly, who was the first African-American woman to get a PhD in chemistry in the United States. And she spent a lot of her career at the Albert Einstein College of Medicine. And I just want to highlight her contributions because, well, let's face it, she's a woman of color, and a lot of times these people have not been recognized the way they should have been throughout the history of science. So I think it's worth pointing out that, you know, biochemistry is open to everybody, right? And when I talk about the Nobel Prize, the fact is that most of the Nobel Prize winners look like me. They're white men. And I'm going to show Nobel Prize winners throughout the course. And I do that just for historical interest. But I don't want you to get the impression that it's only white men who can do important things in science, right? Because obviously that's not true. And so here's an example then of someone who's not a white man who accomplished something pretty significant, right? In the work that she did with her colleagues was to determine the link between high cholesterol and high blood pressure. And this is, you know, research is carried on, and we now accept this as sort of a given. But, you know, obviously there was a time when nobody knew that. And so that her team helped sort this out. So I just wanted to call attention there to someone who knew a lot about steroids during her career. Okay, and then finally, we're going to end up with another class just called other, and these are sort of miscellaneous things that don't fit into the other classes that I've shown, and there's a wide variety of different possible structures. And one example is this, retinol, also you know, related to vitamin A. So vitamin A structure is pretty much like this, retinol. This is not a fatty acid, so there are trans double bonds in this. Again, you have a lot of trans double bonds in lipids, but there's this molecule is synthesized differently than fatty acids are. And don't worry about that. And so anytime you see a structure of a lipid that doesn't fit into one of those other categories, we'll just call it, you know, other lipids. So retinol is important in vision and other things in development. And it's a pretty important molecule. This is a person who studied retinol, and he looks pretty happy in this picture because he won the Nobel Prize, you know, in 1967 for his work on figuring out retinol, what it does, what its role is, and that sort of thing. Okay, so, you know, and then as another example, prostaglandins have a structure that looks like this. Now this, you see, it's got a bunch of sort of hydrophilic groups on it, but it's predominantly hydrophobic, so it's not going to be very soluble in water. So it's still classified as a lipid. If you have a headache and you take a painkiller for that, chances are it's targeting the production of a molecule like this, because this tends to be involved in pain and inflammation, that sort of thing. And that seems important. So these people won Nobel Prize for their work on that. Biochemists, right? So I'll just leave you with that today. Studying biochemistry can lead you to important discoveries that are honored with the Nobel Prize, especially if you're a white male. Okay, so we'll see you tomorrow in this class. We'll continue topic 6. Oh, well, thank you. That's very nice.